Welcome back, everyone. As always, my name is Ken, and for today, I'm going to talk to you about the difference between subthreshold stimuli and summation of contraction. In past semesters, it seems that these two topics are a little too similar in terminology, so it is important that we do distinguish the differences between them. So let us begin with looking into summation of subthreshold stimuli. When we hear about the term subthreshold stimuli, we should be associating it to cells that can be excitable and give off action potentials and depolarize. You can have this occur in neuronal cells or even muscle cells, among other types as well. So let's assume that this is actually a graph depicting the electrical activity of a neuronal cell. Subthreshold, like submarine, means to be below threshold. Okay? And so if this is our threshold for a neuronal cell, that upon reaching it, it would let off an action potential, then the activity that represents subthreshold stimuli should be anything that occurs below this dashed line. So if something does not reach threshold, as seen in these two cases, we have two EPSPs, or excitatory events, then there is no summation, there is no action potential. What ion is associated to this phenomenon? So if you could remember, an EPSP, which stands for excitatory postsynaptic potential, associates the idea that this neuron is actually in connection or in association with several other neurons, and that they communicate from one to the other. This is going to be the neuron that is receiving information from presynaptic neurons. And that information that it receives, once it is within our target cell, is known as a postsynaptic potential. You can have two types of postsynaptic potentials. They could be excitatory or inhibitory. On screen, what we're supposed to be seeing here are two EPSPs, excitatory postsynaptic potentials. They have not reached threshold, so there is no summation. In this case, two EPSPs are occurring, and they are occurring within a close enough proximity that we do finally have this neuronal cell reaching threshold and then giving off the very iconic action potential signature. So, once again, what ion is associated with this? Well, it's actually sodium, because every time an EPSP occurs within our cells, the cells increase their permeability to sodium. Why is this increasing the internal potential so that it reaches negative 55? Well, if you could remember, there is an abundance of sodium found outside of our cells due to a sodium-potassium pump. Sodium-potassium pumps are a form of primary active transport, and when they are functioning, they use ATP, and every time they actually execute a function, they are pumping out of the cell 3 sodium ions and pumping into the cell 2 potassium ions. So there's a lot of sodium outside the cell, and based on the law of diffusion, where would sodium want to go is back in. When an EPSP happens and the permeability of our cell is increased to sodium, we'll have sodium flooding back into a cell and then increasing its potential and getting it closer and closer to, in this case, negative 55. To conclude, if we have two EPSPs within close enough proximity of time, which is a form of temporal summation because temporal refers to time, we end up increasing the amount of sodium that accumulates inside of our neuronal cell, allowing it to reach threshold and then fire off an action potential. So the ionic basis of summation of subthreshold stimuli is simply that sodium is accumulating within the cell. Now, how is this different from summation of contraction? Well, when you think about contraction, what you should be thinking about is, hey, what kind of cell actually could contract, like an accordion? And the first cell that should come to mind, especially as we proceed through this part of our semester, is muscle cells. because of the contraction of muscle cells, we can actually move our body, that's skeletal muscle, and we can also move parts of our organs like our heart and our diaphragm through contractile events. So the terminologies like subthreshold stimuli and contraction should be giving you a hint as to what context we are looking in. So, summation of contraction. You may already know what a twitch is. We say that a twitch is the resulting contractile event following one stimuli. If we end up increasing the frequency of stimulation, now this action potential is what we associate to something that a neuron is probably firing off. Remember, this is a muscle cell, so if a neuron is firing an action potential upon it, that's going to be at what we call a neuromuscular junction. In the previous slide, when we look at summation of subthreshold stimuli, the stimuli that are increasing the internal potential of our neuron cell is not an action potential, those are EPSPs. In this case, it is a muscle cell that we're looking at, and the stimulus is in the form of an action potential coming from a neuron. Their communication is at our neuromuscular junction. So, if we increase the frequency in which action potentials are being fired, seen here and here, then we start exhibiting a form of summation. Right? And because these twitches are building upon each other and they are still visible, then we call this incomplete summation. You see it here as well. It is also known as TREP, and TREP is German for staircase, which is why this kind of looks like a staircase, and that's where it got its name. So this is incomplete summation, also known as TREP, and also known as, what was it? I can't remember. Oh yeah, unfused tetanus, that's the word. Okay, so the only difference between the twitch and TREP is frequency of action potential generation. On an ionic basis, what is happening? The ions that are necessary to get summation of contraction are specifically calcium. Calcium, anatomically, is located within muscle cells inside of the sarcoplasmic reticulum. Muscle cells are unique because their endoplasmic reticulum is modified into what we call the sarcoplasmic reticulum, and it is a very fancy storage unit for our calcium. Every time an action potential is fired off, we end up having those cells release a little bit more calcium, and that calcium enters sarcomeres within the muscle cell and helps those sarcomeres engage in contraction, which we call excitation-contraction coupling. Once frequency of stimulus from action potentials is high enough, we get complete summation, fused tetanus, or tetany. That's what we call this. So, to conclude, the ionic basis for summation of contraction is calcium accumulation within our muscle cells, specifically addressing sarcomeres. And when two action potentials are close enough together, as seen in this case, frequency increases between action potentials, we see that muscle contractions begin to fuse and then lead to TREP and then finally tetany. That should help clear up the difference between summation of subthreshold stimuli and summation of contraction. Thank you for listening.